Ever get that feeling, like when a certain smell brings back a whole flood of memories?
Oh, absolutely, like Proust and his Madeleines.
Exactly. It's like our brains are hardwired to make these lightning-fast connections. Well, this year's Nobel Prize in Physics celebrates the brilliant minds who figured out how to recreate that kind of associative memory in a computer.
It's remarkable, isn't it? The work of John Hopfield and Geoffrey Hinton really bridged this gap between how our brains work and how we can make machines that learn.
It's mind-blowing. So let's start with the first pioneer, John Hopfield, and this thing he created back in the 1980s called the Hopfield network. What is that exactly? It's not your typical computer program, is it?
Not at all. Imagine, if you will, a network of light switches, but on a massive scale. And each switch represents a tiny piece of information.
Okay, I'm picturing a room full of light switches, like something out of a sci-fi movie.
Haha, well, sort of. Now, imagine that these switches are all interconnected and can influence each other. That's the fundamental idea behind a Hopfield network.
So instead of storing information in a specific location, like we do on a hard drive, it's more about the connections between these light switches.
Precisely. Think of it like a spider web covered in tiny dewdrops.
Okay, spider web. I like where this is going.
So each dewdrop might represent a memory, and the web itself is the network. Now, if you were to gently touch the web, what happens?
The whole thing vibrates.
Yes, and the dewdrops closest to your touch, they jiggle the most, right?
Right.
That's analogous to how a Hopfield network retrieves information. You present it with an input, maybe a partial or distorted memory, and the network activates the dewdrops, the nodes that are most closely associated with that input.
So it's not remembering in the way we typically think of it. It's more like it's reconstructing the closest memory based on these interconnected nodes activating each other, like a chain reaction.
Exactly. And the beauty of it is that even with incomplete information, the network can still converge on the most relevant memory.
That's incredible. But I have to ask, how on earth did Hopfield even come up with this idea? Was he trying to build a brain from scratch?
Well, believe it or not, his inspiration came from a rather unexpected source: magnets.
Magnets, you mean those things we stick on our fridges?
The very same. You know how magnets have a north and south pole and they either attract or repel each other?
Yeah, basic physics.
Well, Hopfield had this brilliant insight that he could represent information in a similar way. By using the principles of magnetism, he realized he could create a network that could store and retrieve information based on the interactions between these magnetic-like elements.
So he basically took the physics of magnetism and applied it to how computers store information. That's some serious genius level stuff.
It was a truly groundbreaking idea. And what's even more amazing is that this concept, inspired by physics, laid the very foundation for the development of artificial neural networks, which, as we know, are the backbone of modern AI.
Wow. Who knew that those little magnets on our fridges held the key to, well, a future powered by AI? This is incredible. But before we dive into that future, let's talk about Geoffrey Hinton, the other mastermind behind this Nobel Prize, and how he took this whole AI thing to another level. Okay, so we've got John Hopfield using magnets to make computers think, well, kind of like our brains do. But then comes Geoffrey Hinton, the other big name in this Nobel Prize story. He's often called the godfather of deep learning, which sounds pretty intense. What did he do that was so different from Hopfield?
You see, Hinton's work was revolutionary because he cracked the code on how to teach computers to actually learn. That's a big deal because it's what paved the way for the AI we're surrounded by today.
Okay, teaching a computer sounds complicated. How do you even begin to explain something like learning to a machine?
Think about it like this. Imagine you're teaching a kid to ride a bike. You could try explaining the physics behind it, the balance and the pedaling and all that, but at the end of the day, they learn by actually doing it.
Falling off a few times, I imagine?
Exactly. By trying, failing, adjusting, and trying again. That's kind of how Hinton's big breakthrough, the Boltzmann machine, works. It's all about letting the machine learn through this kind of trial and error process.
So instead of us humans programming in all the rules, Hinton's Boltzmann machine is figuring things out on its own.
You got it. And here's where the physics comes in again. Hinton's work relied on a field called statistical physics.
Statistical physics, huh? That sounds a little intimidating.
It might sound complicated, but it's really about understanding how systems with tons and tons of interacting parts behave. Think of like a pot of boiling water.
Okay, I can picture that. Lots of bubbles, lots of heat.
Right? Now, you can't track the movement of each and every water molecule in that pot, can you?
Not a chance. Way too chaotic in there.
Exactly. But you can understand how the water behaves overall, temperature, pressure, the way it boils, all that comes from the collective movement of all those water molecules. Statistical mechanics helps us analyze that.
So instead of programming every tiny rule into the AI, Hinton's Boltzmann machine is like analyzing a ton of data and figuring out the patterns hidden within it, just like we figure out the overall behavior of the boiling water.
Bang on. It's like the machine is searching for the secret recipe behind all that data, the underlying principles that make it all tick.
Okay, I think I'm starting to get it. But how is this different from Hopfield's network? They both seem to be about recognizing patterns in a way.
The key difference lies in what we call hidden layers. Think of a detective solving a case, right? They don't just look at the evidence lying around and call it a day. They use their knowledge, their experience, their gut feeling to connect the dots and uncover the hidden relationships between those clues.
So are you saying that these hidden layers are like the detective's thought process, analyzing the data and finding connections that aren't obvious at first glance?
That's a fantastic way to put it. These hidden layers are what allow the Boltzmann machine to learn patterns that are much more complex, much more abstract. It's not just about recognizing a cat in a photo anymore. It's about understanding what makes a cat, all those subtle features and behaviors.
Wow, so we've gone from simple pattern recognition to like a deeper understanding of the data. That's a huge step.
Absolutely. And this breakthrough, this idea of adding hidden layers and using statistical physics, this is what opened the door for deep learning algorithms, the stuff that powers today's most advanced AI.
Hinton really changed the game then. From magnets to boiling water to super sleuth AI, it's amazing how it all connects. But enough about the science for now. In the final part of our deep dive, let's bring this home and explore how these incredible discoveries impact our daily lives in ways we might not even realize. Okay, so we've covered a lot of ground here. Magnets, boiling water, detective AI. It's amazing to think that it all started with physics, and now it's shaping the future of, well, pretty much everything. But let's bring this down to earth for a minute. How do these brainy breakthroughs actually show up in our everyday lives?
Oh, they're everywhere. Think about those uncannily accurate recommendations you get on streaming services. Like you watch one documentary about underwater basket weaving and suddenly you're bombarded with suggestions for obscure documentaries about 18th century cheesemaking.
Yeah, it's a little creepy sometimes, not gonna lie, but also cool. How does that even work?
Neural networks. They're trained on huge, massive data sets of viewing habits. And they get really good at predicting what you'll like based on, well, patterns that even you might not realize are there.
So it's like having a mind-reading personal assistant, except it's not reading your mind, it's reading the minds of, millions of other viewers.
Pretty much. And that same technology, those same principles, they're being used in all sorts of fields. Take medical diagnosis, for example.
Oh, right. I've heard about AI being used in healthcare.
Imagine a doctor looking at a patient's scans, right? Now, picture that doctor having an AI assistant that's already analyzed millions of other scans, learned to pick up on the tiniest anomalies, things that even a trained human eye might miss.
So it's like having a second opinion, but a super-powered second opinion. All thanks to this Nobel Prize winning science we've been talking about.
Precisely. And it goes beyond healthcare. Self-driving cars, they rely heavily on these neural networks to understand their surroundings in real time. It's essentially Hinton's vision of machine learning in action, taking in a constant stream of data and making split-second decisions, except this time, it's happening on 4 wheels.
Wow. Okay, so from helping us choose a movie to, preventing accidents on the road, that's a pretty big impact.
It really highlights how these abstract ideas from physics have completely revolutionized the way we live our lives. And it's not just about convenience or entertainment either. It's also about pushing the boundaries of scientific discovery itself.
Now that you mention it, I read about AI being used to analyze data in astronomy and climate science and all sorts of complex fields.
It's true. Think about it. Researchers are using neural networks to sift through these mountains of data, looking for those hidden patterns and connections that would take humans, lifetimes to uncover on their own. It's like having a superpowered research assistant that can process information at warp speed.
So we've gone from recommending movies to potentially unlocking the secrets of the universe. That's incredible.
It's a little bit mind-boggling, to be honest.
It's definitely an exciting time to be alive. And as we venture further into this uncharted territory, this world of artificial intelligence, one thing's for sure, the journey has just begun.
And that's a wrap on our deep dive into the 2024 Nobel Prize in Physics. We've journeyed from the mind-bending world of quantum mechanics to the complex workings of neural networks, and we've seen how two brilliant minds, John Hopfield and Geoffrey Hinton, took inspiration from the fundamental laws of physics to shape the future of artificial intelligence as we know it.
It's been a pleasure joining you on this exploration. And for our listeners, as you marvel at the wonders of artificial intelligence in your everyday lives, remember the incredible journey, the fusion of physics, computer science, and human ingenuity that made it all possible. All right, everybody, buckle up, because we're diving into some seriously cool science today. You sent in your research on this itsy-bitsy worm, C. elegans.
And before you think, worms, boring.
Trust me on this. This little critter holds the secrets to a huge e-discovery, micro-NA. We're talking Nobel Prize winning science here.
It really is remarkable how much we can learn from something so small. C. elegans is like having a living, see-through textbook for genetics.
You got that right. Scientists adore these worms. They're practically invisible under a microscope, and their life stages are predictable, like clockwork. But here's where things get really mind-blowing. Today, we're zeroing in on two genes, Lin4 and Lin14.
They basically act like the gas pedal and the brakes for this worm's development. But you guessed it, when you start messing with those genes, things get a little funky. Think of it like a beautifully choreographed dance, with these genes calling all the steps, get the steps wrong, and the whole routine goes haywire.
Okay, so walk me through the dance, the life cycle. What are the usual steps these worms go through?
So baby C. elegans, or larvae as they're called, they go through 4 main stages as they grow up, kind of like levels in a video game. We're talking L1 to L4.
I like it.
And each stage, well, it has these very specific developmental milestones, things that have to happen before the worm can move on to the next level, you know.
Right, makes total sense. But what happens with those worms we talked about, the ones with the messed up genes, the mutants?
Yeah.
You've got the ones labeled Lin4 and Lin14-0.
Okay, so the Lin4 mutants, it's like someone hit the repeat button on that first stage, that L1 stage.
They keep molting, shedding their skin, but they never really level up to those later stages of the game. They don't develop the right adult features, can't lay eggs. It's like they're stuck in a never-ending childhood.
Whoa, that's nuts. So they're like the Peter Pans of the worm world.
But going in the total opposite direction, you've got the Lin14-0 mutants.
Those guys are like the teenagers who just can't wait to grow up. They blaze through their larva stages, skipping steps, jumping straight to adulthood.
But I'm guessing it's not all sunshine and roses for those speed demons. Gotta be some downsides to rushing through development like that.
You're spot on. Because as fun as it might sound to skip that awkward teenage phase, these Lin14-0 mutants, they end up with problems, developmental problems, missed a few too many steps along the way, right? They might not form all the right tissues or organs. Their whole development is just off.
Fascinating. So we've got Lin4 pumping the brakes and Lin14 slamming on the gas. And by studying what happens when these genes go haywire, scientists can start to unravel this mystery, how developmental timing works in these worms.
Exactly.
Which, like you said, could have much bigger implications down the line.
Absolutely. And this is where the real breakthrough comes in. Now, remember how we've been talking about genes and how they usually do their thing by making proteins?
Well, Lin4 decided to throw a curveball. Researchers, they were expecting to find some protein, right? Some protein that interacts with Lin14, but guess what? Nothing. They found nothing.
Wait, back up. You're telling me this Lin4 gene, this key player, it doesn't even make a protein?
Nope.
That's like saying the engine in my car runs on rainbows and good vibes. How does a gene even work without whipping up a protein?
That's the million-dollar question, isn't it? Turns out, Lin4 had a secret weapon.
Something completely unheard of back then. Tiny pieces of RNA. They called them Lin4S and Lin4L.
Okay, now you're speaking a different language. We talked about genes, we talked about proteins, but RNA, I haven't heard that word since high school bio.
All right. Think of it like this DNA. It's the master blueprint, right? Holds
Yeah.
all those genetic instructions. But DNA's got an important job, so it stays locked up tight in the nucleus.
Okay.
RNA, on the other hand, that's like the working copy of those instructions. Carries the genetic code from DNA out to the ribosomes.
And that's where proteins are actually made.
So these Lin4S and Lin4L things, these snippets of RNA, they're like messengers gone rogue. What are they doing if they're not making proteins?
I wouldn't say rogue, not exactly. They're more like highly specialized editors.
See, instead of carrying the code to build something, these tiny RNAs, they silence the message of other genes. Remember that Lin14 gene, the one that's basically flooring it?
Oh yeah, the speed demons.
Right. So Lin4S and Lin4L, they latch onto the Lin14 messenger RNA, like slapping a piece of tape right over a barcode, you know what I mean? Ribosome comes along, can't read the code, no Lin-14 protein is made.
Wow. Talk about sneaky.
Mm.
These tiny RNAs are slapping a gag order on Lin-14, stopping it from speeding up development. And this was brand new information back then.
Brand spanking new. They call it antisense regulation. Those tiny RNAs were binding to the opposite strand of the messenger RNA, the antisense strand.
It was a huge discovery,
Yeah.
totally revolutionized how we think about gene control.
Mind officially blown. So it's not just genes making proteins talking to each other. We've got this whole other layer of control going on with these tiny RNAs calling the shots, like uncovering a whole secret language inside our cells. But was this RNA silencing thing just a fluke, something unique to these worms?
Well, that's the thing. Remember Lin-4, the one making those tiny RNAs?
Yeah.
It had a distant cousin, a gene called Let7. And guess what?
Let7 was making microRNAs too.
Okay, so another gene making these tiny RNAs. What's the big deal? Okay, so another gene making these tiny RNAs. What's the big deal with this Let7 character? Is it just more of the same, keeping some other gene in check?
Here's where things get really wild. Let7. It wasn't just controlling some random process in the worm.
It was doing something fundamental to development.
Okay.
Okay, this Let7 wasn't just hanging out in worms.
Researchers found it in flies.
Fish, mice, even humans.
Whoa, hold the phone. You're telling me this tiny piece of RNA,
This microRNA.
it's doing something important in all these different creatures. We're talking like a universal language of gene regulation here.
Exactly. That's what blew the field wide open. Let7 showed everyone this microRNA system.
This wasn't just some weird worm quirk.
It was a fundamental mechanism of gene control.
Conserved across millions of years of evolution.
So this is way bigger than just figuring out how a worm develops. We're talking about maybe understanding this hidden layer of regulation.
In our own cells.
Yeah.
But you said humans have thousands of these microRNAs. What are they all doing?
That's the million-dollar question.
The scientists are working hard to figure that out.
We're discovering new microRNAs all the time, and they're involved in so many processes.
Everything from embryonic development to immunity to brain function.
Okay, so we've got these tiny RNAs acting like puppet masters.
Pulling the strings of gene expression in all kinds of cells. This is all incredibly cool, but I have to ask, what's the practical side of all this? How does understanding microRNA help us, say, cure diseases or develop new treatments?
Okay, well, think of it like this. MicroRNAs, they're the conductors of this cellular orchestra.
And diseases, things like cancer, heart disease, even neurological disorders.
They're like disharmonies in the music.
The wrong microRNAs are playing too loud, others are too quiet, and the whole symphony falls apart.
So messed up microRNAs could be at the root of all sorts of health problems.
Exactly, and that's what makes this research so exciting.
What if we could develop treatments that target these microRNA malfunctions?
Imagine being able to silence those overactive microRNAs driving a tumor's growth.
Or boost the activity of microRNAs that have gone quiet in a neurodegenerative disease.
It's like having a volume knob for our genes.
Fine-tuning their activity to restore order in the body. We could be talking about a whole new era of personalized medicine. This is incredible.
It really is. And while we still have a lot to learn,
The potential of microRNA-based therapies, it's enormous.
we're already seeing promising results in clinical trials for cancers and other diseases.
So from a tiny worm to potentially revolutionizing medicine, who would have thought?
It just goes to show every discovery, no matter how small or unexpected,
It has the potential to unlock something incredible. Who knows what amazing breakthroughs are waiting for us just around the corner.
that's the beauty of science, isn't it? This deep dive has been a wild ride, and I, for one, can't wait to see what the future holds for the world of microRNA.
Get ready to explore the world of proteins. They're like the tiny building blocks of life. And you know what's amazing? Scientists are figuring out how to build these proteins from scratch.
Yeah, it's like having the power to write the blueprints of life itself.
Exactly. We're going to unpack how this groundbreaking work earned the 2024 Nobel Prize in Chemistry.
It's a story that unfolds like a detective novel, full of puzzles, breakthroughs, brilliant minds, the whole shebang.
I love a good science mystery. So where do we even begin?
Okay, so imagine proteins like incredibly intricate origami.
Okay.
Each fold has to be precisely right for the protein to do its job.
Okay, I can picture that. So a protein's shape is its destiny. How did scientists even begin to unravel these microscopic structures?
Well, way back in the 1950s, scientists used this technique called X-ray crystallography.
Oh,
It's like taking a super high-resolution X-ray of a molecule to reveal its 3D structure.
oh, so they were finally able to see these intricate folds in all their glory.
Exactly. It was revolutionary. Two scientists, Kendrew and Pritz, won the 1962 Nobel Prize in chemistry for using this technique to reveal the first ever protein structures. It was like opening a window into this hidden world.
That's a huge leap forward.
Yeah.
So what's next in our protein saga?
Well, then came a scientist named Christian Anfinsen. He discovered something truly remarkable. A protein's amino acid sequence, so the specific order of its building blocks, actually determines its 3D shape. Think of it like this: hidden within the protein's building blocks is a code that dictates the final folded structure.
Okay.
This discovery earned Anfinsen the 1972 Nobel Prize in chemistry.
Wow, so the sequence is like a set of instructions that the protein follows to fold itself up.
Precisely. But here's where it gets really interesting and a bit mind-boggling. A protein could theoretically fold in an astronomical number of ways. We're talking more possible shapes than there are atoms in the universe.
Oh, wow.
It's like trying to fold a piece of paper into a swan, but there are trillions upon trillions of possible swan-like shapes.
That's a lot of pressure for a tiny protein. So how on earth does it find the right shape so quickly?
That, my friend, is known as Levinthal's paradox.
Okay.
It suggests that proteins don't just randomly fold until they stumble upon the right shape. There must be some guiding principles, a sort of protein choreography that we haven't fully deciphered.
Okay, now I'm really curious. What does this have to do with the 2024 Nobel Prize?
Anfinsen's work raised a critical challenge. Could we predict a protein's 3D structure just by looking at its amino acid sequence? Cracking this protein folding code would be a game changer.
Yeah, it'd be like having a superpower, the ability to glance at a protein's building blocks and know exactly what it looks like and how it functions. But how do you even begin to solve a problem that complex?
That's where the CASP competition comes in. It stands for Critical Assessment of Protein Structure Prediction.
Oh.
It became the battleground for the brightest minds in the field.
Okay, I'm picturing scientists facing off in a giant auditorium. Protein structures projected on a screen. Like, give me the details. What went down at these protein prediction showdowns?
You're not far off. At CASP, scientists would use their algorithms to predict the structure of a protein and then compare their predictions to the actual structure, which was determined experimentally.
So it's like a science bake-off.
Kinda, yeah.
Whose algorithm could bake the most accurate protein structure?
Exactly. And for many years, progress was steady but like incremental, you know? Then in 2018, a team from DeepMind, led by Demis Hassabis, shook things up.
Wait, Demis Hassabis, the chess prodigy turned AI master.
Yeah.
Wasn't he the one behind AlphaZero, the program that conquered the game of Go?
The very same.
Oh,
Hassabis realized that the same AI techniques used to master those complex games could be applied to the protein folding problem.
That's fascinating. It seems like everything's connected. But how do you even begin to teach an AI to understand the intricate folds of a protein? That's like next level stuff.
They used a type of artificial intelligence called a convolutional neural network.
Okay.
Imagine a computer program that learns to recognize patterns by looking at thousands of images.
Oh.
That's essentially how AlphaFold was trained. It analyzed a vast database of protein structures, learning the relationships between amino acid sequences and their corresponding 3D shapes.
So it's like showing a child a bunch of different origami animals.
Right.
And then giving them a new set of instructions to fold. They start to see the patterns and figure out how the folds create the final shape.
That's a great analogy, and it worked brilliantly. In the 2018 CASP competition, AlphaFold blew the competition out of the water.
Okay, spill the tea. Like,
Yeah.
how much better was AlphaFold than the other contenders?
Well, scientists use a metric called the GDT score to measure the accuracy of protein structure prediction.
Okay.
A score of 90 or above is considered as good as solving the structure experimentally.
Okay.
Before AlphaFold, the best programs were only achieving GDT scores in the 60s.
60%, that's like passing, but not exactly stellar.
Yeah.
what did AlphaFold score?
AlphaFold achieved a GDT score of around 60 as well, which was still very impressive for the time.
Right.
But the DeepMind team, they weren't finished. They brought in a physicist named John Jumper, who helped them take AlphaFold to a whole new level.
Okay, John Jumper sounds like a critical character in our protein story.
Yeah.
What did he bring to the table?
So Jumper was already renowned for his work on simulating protein dynamics, basically how proteins move and interact over time. He realized that while AlphaFold was revolutionary, it could be even better.
Oh, wait.
So they revamped the entire program.
So it was a complete protein makeover. What did this, AlphaFold 2.0 or whatever they called it look like?
They called it AlphaFold 2.
Okay.
And it incorporated some cutting-edge AI techniques, including something called transformers.
Transformers?
Yeah, imagine transformers as pattern recognition on steroids.
Okay.
They're really good at understanding long-range relationships in data, which is crucial for protein folding.
Okay, so transformers can see the big picture.
Yes.
Understanding how one part of an amino acid sequence affects a distant part of the folded protein.
That's a great way to put it. But they didn't stop there. They also implemented this ingenious approach called the residue gas method. Imagine each amino acid as a tiny particle, floating around in a simulation.
Okay.
AlphaFold 2 would use algorithms to move these particles around, testing different configurations until it found the most stable and energetically favorable structure.
So they basically recreated the protein folding process inside a computer,
Yeah.
letting the AI figure out the most likely structure.
Precisely. It was a stroke of genius.
Yeah.
And the results, let's just say AlphaFold 2 completely dominated the CASP14 competition.
I bet they were probably popping champagne corks left and right.
Give me the numbers. What kind of scores did AlphaFold 2 achieve?
Get this. AlphaFold 2 achieved a GDT score of around 90%, essentially matching the accuracy of experimental methods. It was a game changer. Wow, they went from a passing grade to acing the exam. That's Nobel Prize worthy for sure. But there's another player in this story, right? We haven't forgotten about David Baker.
Absolutely not. While AlphaFold was busy cracking the code of protein prediction, David Baker was taking a different but equally revolutionary approach.
Yeah, okay.
He was focused on protein design, building entirely new proteins from scratch.
Whoa, hold on a minute. Predicting protein structures is one thing, but designing them, that sounds like something straight out of science fiction. How is that even possible? Designing a protein, it's like composing a symphony, but instead of notes, you're arranging amino acids. So where does one even begin?
So David Baker started with the same fascination with protein folding as Hassabis. His team created this software program called Rosetta.
Okay.
Initially, it was aimed at predicting protein structures.
So Rosetta was like an early prototype for AlphaFold.
You could say that, but here's where Baker's ingenuity really shines through. He realized that if Rosetta could predict a protein shape from its sequence, maybe, just maybe, they could use it in reverse.
Okay, now my mind is officially blown. They were trying to reverse engineer the blueprint of life.
Precisely. Imagine trying to deduce the exact sequence of folds needed to make an origami crane. But instead of paper, you're dealing with molecules.
Right.
It was a long shot, but they fed Rosetta hypothetical protein structures and challenged it to generate the amino acid sequences that would fold into those shapes.
And what happened? Did it work?
It did.
Wow.
In 2003, they created Top 7, a protein with a completely unique structure never seen before in nature.
Wow.
And the best part, it folded exactly as Rosetta predicted. It was a resounding confirmation that protein design was not just a distant dream, but an emerging reality.
That's incredible. It's like they discovered a new element.
Yeah.
But instead of the periodic table, it's the protein building blocks of life. So what were some of the other amazing things Baker's team created with this technology?
Oh, Baker's lab became a hotbed of innovation. They designed proteins that could assemble themselves into tiny cages.
Really?
Like microscopic delivery vehicles for drugs. They even created proteins that could act like molecular rotors, spinning around like tiny machines.
Wow, that's like having a nanoscale toolbox.
Yeah.
So what are some of the potential applications of this technology in the real world?
Oh, the possibilities are truly vast. Imagine designer proteins that could precisely target cancer cells, delivering drugs with pinpoint accuracy and minimizing side effects.
Wow.
Or proteins engineered to break down plastic waste, offering a solution to, one of our planet's most pressing environmental challenges.
It sounds like protein design could revolutionize medicine, material science, environmental science, the list goes on. And with AlphaFold 2 being open source, making this knowledge accessible to scientists worldwide, the pace of discovery is only going to accelerate.
Absolutely. We're living in an incredibly exciting time for protein science. We've gone from like struggling to determine the structure of a single protein to predicting and even designing them with incredible precision.
Right.
It's a testament to human ingenuity and the power of scientific collaboration.
It's truly mind-boggling. We've covered a lot of ground in this deep dive, from the early days of X-ray crystallography to AI-powered protein design. We've seen how scientists are unraveling the mysteries of protein folding and using this knowledge to engineer solutions to some of the world's biggest challenges.
Yeah.
It really makes you appreciate the incredible complexity and elegance of the natural world and how much more there is to discover.
It really does. And it begs the question, if we can now read and even write the blueprints of life, what will we choose to build?
All right, so have you ever finished a book and just thought, wow, I need to process that?
You just like stare at the ceiling for a bit?
Yes,
Yeah.
exactly. You know those books that really stay with you? Well, let's talk about Han Kang, the winner of the 2024 Nobel Prize in Literature. Because her books, they're exactly that kind of powerful.
I think a lot of people are going to start hearing her name everywhere now.
Definitely. So to really dive deep, no pun intended, we're focusing on the Nobel Prize announcement itself and a recent profile on Han Kang from The New Yorker. Kind of get a sense of what she's all about.
Those are great sources for this.
Definitely. And, you know, the Nobel Prize, that's like the peak of achievement for an author.
That's a huge honor. Huge.
Right. Like the whole literary world is watching. So when they make their choice, people pay attention. And for Han Kang, they said she won for her intense poetic prose that confronts historical traumas and exposes the fragility of human life.
Phew.
That's a mouthful, but there's so much to unpack there.
It's a really powerful statement. I think the first thing that jumps out is that phrase, historical traumas, because it signals right away that her work is dealing with some heavy stuff.
It makes you wonder, what exactly are those traumas?
Right. For Han Kang, a lot of it stems from Korean history, especially the Korean War. I mean, that war was devastating. It tore the country apart, literally and figuratively, and the effects of that division are still felt today, both in North and South Korea.
That's true. We often think of historical events as being, well, in the past. But Han Kang's work seems to show how deeply those events can resonate through generations.
Exactly. And she doesn't shy away from those difficult emotions. That's where I think the intense poetic prose comes in. Her writing is very visceral. She uses these incredibly vivid images and sensory details that pull you right into the experience of her characters.
You almost feel like you're living it alongside them.
Yes. And we know one example that always comes to mind is her novel Human Acts. Have you read that one?
I have. It's tough, but incredible.
It's about the Gwangju Uprising, which was this brutal crackdown on a pro-democracy movement in South Korea in 1980. And the way Han Kang writes about it, you feel the chaos, the fear, the violence, but also this incredible resilience of the human spirit.
It's definitely not an easy read, but it's so important. And I think that's one of the things that makes her work so compelling. She tackles these really tough subjects head on, but she does it with such beauty and empathy.
Absolutely. And you know, what's interesting is that while her stories are deeply rooted in Korean history and culture, the themes she explores are actually universal.
Oh, for sure. Trauma, loss, resilience, the search for meaning and connection in a chaotic world. These are things that everyone can relate to on some level.
Exactly.
And that's where I think this Nobel Prize is so exciting. It's not just an award for Han Kang, but it's an opportunity for readers around the world to discover her work and connect with these powerful stories.
It's like opening a window to a different part of the world.
Exactly. So if you're looking for a book that will challenge you, move you, and stay with you long after you've finished the last page, I highly encourage you to check out Han Kang's work. And maybe, just maybe, it'll spark a new curiosity about Korean history and culture. You might even find yourself reflecting on how these events, though worlds apart, might echo in your own life. It's incredible how literature can do that, right? It bridges those gaps and makes us realize just how interconnected we all are.
Yeah, it's amazing.
So I guess a good question for everyone listening might be this. How has reading about historical events, maybe even ones far removed from your own experience, changed how you see the world, or even yourself? Think about it, and maybe pick up a Han Kang book. You won't regret it.